Hello students, welcome to Biology CET class. Today's topic is on anatomy of flowering plants. Before solving questions, let me briefly explain the chapter. So what is anatomy? Anatomy means it is a study of internal structure of a plants. What are all present in the internal structure? Internal structure contain a cell. That is, cell is a basic unit of a living organism. Those that cells are organized into tissues, and in turn the tissues are organized into organs. Different organs in a plant show difference in their internal structure. So within the angiosperm, monocot and dicot are also seen to be anatomically different. So the internal structure depends on the adaptation to diverse environments. So tissue is nothing but a group of cells having a common origin and usually performing a common function. Tissue is divided into two types, meristematic and permanent. Meristematic means the cells are actively they divide further and further. But in the permanent, where the cell divide only once, there is no further division. This is about the types of tissues. Next is the types of meristematic tissue. Meristematic tissue has two types, primary meristem, secondary meristem. Again, primary meristem is subcategorized into intercalary meristem, apical meristem, secondary meristem into lateral meristem. Apical meristem again divided into shoot apical meristem, root apical meristem. Let us study one by one. Primary meristem has two types, apical meristem and intercalary meristem. Apical meristem means meristem occur at the tip of the shoot and root and produce primary tissue called apical meristem. If it is present at the shoot tip, it is called shoot apical meristem. If it is present at the root, it is called root apical meristem. You can observe here, this tip is called the apical meristem. Next is the intercalary meristem. The meristem occurs between mature tissue. It is called intercalary meristem. Intercalary meristem helps in the increase of the length of the plant. So in this region, intercalary meristems occurs. Next is secondary meristem. Secondary meristem type is lateral meristem. Meristem occurs at the mature regions of the root and shoot of plant. It increases the girth of the plant. Example for the lateral meristem is fascicular vascular cambium, interfascicular cambium, core cambium and intrafascicular cambium. These are the example for lateral meristem. Lateral meristem helps in the increase in the girth of the plant. So in this region, the lateral meristem occurs. And the next is permanent tissue. Till now we studied about the meristematic tissue and its types. What is permanent tissue? It is divided into simple tissue and compound tissue. Simple tissue means it has only one type of cell. Means all cells are similar. It is made up of only one similar cell. Whereas compound tissue, more than one type of cell. That is, it is composed of more than one cell. It doesn't have a similar cell. Whereas simple tissue, again it is divided into three types, parenchyma, collenchyma, sclerenchyma. Compound tissue, it is again divided into two types, xylem and phloem. Let us study now simple tissue. See, under the simple tissue, parenchyma cells is present. So parenchyma cell has a dense cytoplasm. It has a dense cytoplasm. If you look at the diagram, you can notice it has a dense cytoplasm and a large vacuole and nucleus. Hence it is a living cell. The shape of the cell is spherical, ovule, round, polygonal or elongated shape, and it is isodiametric. Isodiametric means all cells are in the same size, and cell wall is very thin and it is made up of cellulose. They may be closely packed or with a intercellular, so you can observe intercellular space with a small intercellular space or without intercellular space. Main function of the parenchyma is storage, photosynthesis and secretion. The next is the collenchyma. Collenchyma is a type of simple tissue and it has a dense cytoplasm. You can observe it has a dense cytoplasm in the picture and it has a large vacuole and nucleus, hence it is a living cell. Collenchyma may be homogeneous or heterogeneous. Homogeneous means it is only made up of collenchyma. Heterogeneous means collenchyma present with the other cell, like it may be parenchyma or sclerenchyma. And it is also isodiametric, means same size. All cells are in the same size and it may be spherical, ovule, round, polygonal or elongated shape. And the cells are thickened at the corners. If you observe at the corners, the cells are thick and which is made up of cellulose, hemicellulose and pectin. Here the intercellular spaces are absent, whereas in parenchyma it may have intercellular space or may not. But in the collenchyma intercellular spaces are absent. Main function of the collenchyma photosynthesis and mechanical support. The next type is sclerenchyma. Sclerenchyma, it is a long narrow with lignified cell wall having numerous pits. This sclerenchyma is a long narrow. If you observe this one, you can notice it is a long narrow with lignified cell wall. At the cell wall, it has a lignin deposition. At some point, it does not, the lignin deposition is not there. That opening is called the numerous pits, and it is a dead end without protoplast. Hence, it is a non-living cell. Function as mechanical support. On the basis of variation in structure, origin and development, it is of two types, fibrous and scleroids. Fibrous, it is thick walled, elongated and pointed. Thick walled, elongated and pointed cells. In the scleroid, it is a spherical, ovule or cylindrical, highly thickened dead cells with narrow cavity, commonly found in fruit walls of nuts, pulp of fruits like guava, pear, sapota, seed coats of legumes and leaves of tea. And the next comes the complex tissue. Complex tissue means made up of more than one cell. So again, xylem composed of four cells. One is tracheids, vessel, xylem fibers, xylem parenchyma. Tracheids, xylem in the tracheids are elongated with thick and lignified walls and tapering ends. These tracheids are elongated with lignified wall. It has a lignified wall. This is what lignified wall, and it has a tapering ends. And tracheids present in a different forms. It may be reticulate, network, annular, etc. Vessel, it is also a long cylindrical tube-like structure made up of many cells, and it is absent in gymnosperm. It is a peculiar character in the angiosperm. So vessel present only in angiosperm. Xylem fibers, next type is the xylem fibers. It is highly thickened walls and obliterated at central lumens. Xylem parenchyma, the name itself indicates it is made up of parenchyma cells, that is living cells. They store food materials in the form of starch or fat and other substance like tannins. Main function of the xylem is conducting tissue for water and minerals from roots to stem and leaves, and it provides mechanical strength to the plant. Next is xylem. Xylem is divided into primary xylem and secondary xylem. Primary xylem is firstly formed, secondary xylem later formed. Primary xylem, again it is divided into protoxylem and metaxylem. So the first form primary xylem is protoxylem, metaxylem later form primary xylem, it is a later form primary xylem. So if you look at the diagram, you can notice The larger one is the metaxylem. So red color, it indicates red color. This is the metaxylem, and the last one, it is the protoxylem. So if the protoxylem towards the center means towards the pith, and the metaxylem towards the endodermis, that is peripheral region, that condition is called endar condition. If the protoxylem towards the periphery and metaxylem towards the pith, this condition is called exarch. This is about the protoxylem and metaxylem. Next is phloem. Phloem is also a type of complex tissue, and it is composed of 4 cells: tube elements, companion cell, phloem parenchyma, phloem fiber. Tube elements, it is a tube-like structure longitudinally arranged with companion cell. Companion cell, it is a parenchymatous cells. It helps to maintain pressure gradient in tubes. Tube elements, it is associated with companion cells. Why? Means tube elements at the maturity time, it lacks nucleus. So what are all the function done by tube elements will be performed by the companion cells, because companion cells are the nucleus. So the nucleus which is present in the companion cells controls the function of tube elements. Next is phloem parenchyma. It is an elongated tapering cylindrical cells which have cytoplasm and nucleus. It secretes food material and other substances like resins, latex, mucilage. Phloem fiber or bast fiber, it is a sclerenchymatous cells. Commonly it is a dead cell. The next is, what is the function of phloem? The function of phloem is transports food material from leaves to all parts of the plant. Again, this phloem is divided into 2 types: protophlloem, metaphloem. So I already said protophlloem is first flea formed primary phloem, consists of narrow tubes. So observe, it consists of narrow tubes. Metaphloem, later formed primary phloem, consists of broad tubes. These are the main 2 difference between protophlloem and metaphloem. The next is tissue system. So till now we studied about the different types of tissues which is present in the plants, how the cells are arranged in the tissue. So on the basis of their structure and location, the tissues are arranged. So this is termed as a tissue system. This tissue system has 3 type tissue system in the plant. So let us see what are all the tissue system present in the plant. First is the epidermal tissue. Second one, ground or fundamental tissue. Third one, vascular tissue. So now one by one we will study. So epidermal tissue, it is a outermost layer. It is a outermost layer. It is outermost layer, and it is made up of parenchyma cells. Parenchyma cells, and within it has a hair-like structure. It is termed as trichome. It is termed as trichome or root hair. And in the root, monocot root, this epidermal tissue, outermost layer, it is termed as epiblemma, and in the dicot it is termed as epidermis. So epidermis in the dicot root, in the monocot root it is termed as epiblemma. And between this epidermal tissue, a kidney-like structure is formed. This is called stomata. This is called the stomata. So let us see the structure of stomata. So stomata in the dicot, it is dumbbell shaped, whereas in the monocot it is in the dicot it is kidney shaped, whereas in the monocot it is dumbbell shaped. And this kidney shaped structure is called the guard cell, and this guard cell is covered by the subsidiary cells. And this guard cell, inside this guard cell enclosed and has a stomatal pore. This is called the stomatal pore, and this region towards the stomatal pore, guard cells are thick, and towards the subsidiary it is thin, because it maintains the turgidity of the cell. That's why it is thick towards the guard cell and thin towards the subsidiary cell. So the main difference between monocot stomata and dicot stomata has a kidney shaped stomata, whereas this monocot, it has a dumbbell shaped stomata. The next is, this is about stomata, and the main function of the stomata is to regulate the transpiration and helps in the gaseous exchange. So regulates the transpiration and helps in the gaseous exchange. Next comes the ground or fundamental tissue. Ground or fundamental tissue present next to the epidermal tissue. This is what a ground or fundamental tissue. Except vascular tissue and epidermal tissue, all tissue present within this tissue system is termed as ground or fundamental tissue, that is cortex, endodermis, pericycle and pith. In leaf, ground tissue is called mesophyll. In leaf, ground tissue is called mesophyll. And the next is vascular tissue. Vascular tissue present within the cortex region. So xylem, phloem is together, it is called the vascular tissue. In dicot stem, xylem and phloem as a cambium. So let us study now the different types of vascular bundles which is present in the monocot and dicot. So vascular bundle is divided into radial and conjoint. Radial and conjoint. In the radial, phloem, xylem present at the different means phloem at one position, xylem at one position, but in the same radii, arranged alternately in the same radii, that is called radial vascular bundle. Conjoint, both xylem and phloem present together in a different radii, that is arranged both. And again this conjoint is divided into conjoint vascular bundle divided into open type, closed type. In the open type, it has a cambium between phloem and xylem. So if cambium present between xylem and phloem, it is called conjoint open type. If it has only one phloem and one xylem, it is collateral. If it has two phloem towards periphery and towards the center, and between that it has a xylem, that is called bicollateral open type. Closed type means there is no cambium between the phloem and xylem, that is called conjoint closed type. Next is, till now we studied about the tissue system and the different types of tissues. Let us now study about the how these tissues are present in monocot and dicot, how they differ in their tissue system. So let us now I'll explain only the difference between the monocot root and dicot root by showing the diagram. So characters, similarities, epidermal cells with root hairs. Both have epidermal cells with root hairs. If you look at the diagram, if you can observe both as epidermal cell with root hair. This is what a root hair. So this is the similarity. Next is parenchymatous cortex is present. So both in the cortex region, in both monocot and dicot, made up of parenchymatous cells. And whereas the next is secondary cortex, the endodermis. Here endodermis is made up of parenchymatous cells, and endodermis as Casparin strips, that is waxy layer. Some of the endodermis as a thickened cells so that it cannot enter a water through this. It is called Casparin strips. It is called a Capstrin strips. And some of these cells does not have this Casparin strips. That cell is called passage cell. Through that cell, the water enters. So Casparin strips is a waxy layer which is present in the dicot root and monocot root. What is the difference? The difference occurs in the vascular bundle. In the monocot it is polyar, in the dicot root tetrad. Xylem and phloem present 4 xylem, 4 phloem present in the dicot root, whereas in the monocot root more than 4. Pith is large and well developed. Pith is large and well developed in monocot root. You can observe pith is large in monocot root and it is conspicuous or small in dicot root. Secondary growth absent in monocot root, secondary growth present in the dicot root. The next comes the stem. We studied now root. So let us see what are all the similarities. Here also epidermis is same in both dicot and monocot stem, and it is covered by a thick cuticle layer. Thick cuticle layer means it is a waxy layer. Thick cuticle layer is a waxy layer. It helps in the breakage of the, it helps to protect the cell from breakage. Hypodermis. Hypodermis is nothing but the cortex region. Here cortex is differentiated into hypodermis and cortex. Hypodermis is made up of collenchyma in dicot stem. Here hypodermis is made up of sclerenchymatous in the monocot stem. Generally the cortex, here the cortex is made up of parenchymatous cells. So this is difference, and major portion of ground tissue is parenchymatous. In both major portion of ground tissue are parenchymatous. Vascular bundle in both dicot system and monocot stem and dicot stem, both vascular bundles are conjoint and collateral. What is the difference? So as I already told, hypodermis in the monocot stem, hypodermis is made up of sclerenchymatous, dicot stem made up of collenchyma. Cortex and stele, the vascular bundles are scattered. If you observe, vascular bundles are scattered. It looks like a human skull in the monocot stem. Whereas in the dicot stem, vascular bundles are arranged in the form ring with distinct cortex and stellar region. You cannot differentiate stellar regions in the monocot stem. Starch sheath absent in the monocot stem, whereas present in the endodermis. Vascular bundles conjoint and closed, whereas vascular bundle is conjoint open endarc. Open endarc means cambius present in the dicot stem, absent in monocot stem. Lysigenous cavity present in the monocot stem, absent in the dicot stem. Phloem parenchyma absent in the monocot stem, present in the dicot stem. Pith not differentiated. Pith is large, made up of parenchyma cell in the dicot stem. So next we will study about the monocot leaf and the dicot leaf. Symmetry dorsiventral in the monocot, dorsiventral in the dicot leaf, whereas in the monocot leaf it is isobilateral, means same side is, both side is same, whereas dorsiventral it is differ in the dorsal side it has a reticulate and less stomata like that. So let us now see stomata equal number on, this is dicotous dorsiventral, monocotous isobilateral. Stomata equal number on both dorsal and ventral surface, whereas in dicot more stomata on the lower surface as compared to the higher surface, because if more stomata present on the upper surface, the transpiration will be more. Bulliform cell present on upper epidermis. Bulliform cell is nothing but a specialized structure. If you look this, enlarged cells, large one, these are the bulliform cells. So this bulliform cells helps the reduction of water in the leaf by curling the leaves. So bulliform cells present in the monocot leaf, in dicot leaf it is absent. Mesophyll differentiated into palisade and spongy parenchyma. In the dicot leaf it is differentiated. So here little bit changes. It is differentiated in the dicot and not in the monocot. And bundle sheath absent in the monocot leaf, present in the dicot leaf. Vascular bundle almost uniform size due to parallel venation, whereas vascular bundle depend on the size of the veins. The next come to the, so if you look at the diagram you can observe, so it has a spongy parenchyma palisade spongy parenchyma are spongy parenchyma are spherical in shape, whereas palisade parenchyma elongated, and palisade parenchyma present in the upper region, upper surface, that is called adaxial epidermis, and spongy parenchyma present in the lower surface, that region is called abaxial epidermis. This is about the leaf. Next comes the secondary growth in dicot stem. What is secondary growth? After the primary growth, some of the cell undergoes secondary growth in the dicot stem. So apart from primary growth helps in the elongation of the plant, that is increase in the length of the plant. So some of the primary cells in the dicot stem, apart from that, even some of the cells increase in the growth of the plant. This increase is called secondary growth, and this increase in the secondary growth are done by lateral meristem. Again, its lateral meristem consists of two meristem, that is vascular cambium, cork cambium. Let us now study about what is this vascular cambium and cork cambium. So if you look at the normal dicot stem, you can notice xylem phloem with cambium, and middle it has a medullary rays. Medullary is made up of parenchymatous cells, whereas this cambium is also made up of parenchymatous cells. This cambium, which is present between the xylem and phloem, it is called interfascicular cambium, and between the vascular bundle it is called intrafascicular cambium. These two inter and interfascicular cambium completely joins to form a ring called vascular cambium ring, cambial ring or a vascular cambium. This is how it occurs. So they divide adjoining. See, if you observe, they divide cambium and medullary rays, they adjoin and start to form a ring called cambial ring or a vascular cambium. After formation of cambial ring, this cambial ring undergo active division, and while dividing, some of the cells cuts off towards inner side and towards outer side. The cell which is cut towards inner side form secondary xylem. It forms secondary xylem. Inner side it is secondary xylem, and the outer it is called secondary phloem. Till we have studied about the formation of vascular cambium. During this formation of vascular cambium and the division cuts off, all this process injures the epidermis and the cortex. So during this division, some of the cell rupture or breakage at the epidermal and cortex region. So this cell should be replaced by some other cell. The some other cell is nothing but the parenchyma cells. That parenchyma cells termed as meristematic tissue, and this meristematic tissue is nothing but a meristematic tissue is nothing but a phyllogen or a cork cambium. This phyllogen actively divides and cuts off cell to the inner and outer side. The outer cell is differentiated into cork or phylum, while the inner cell differentiated into secondary cortex or phylloderm, where this phyllogen, phylum, phylloderm, collectively it is called as a periderm. This is how the cork cambium is formed in the cortex region. So this is the growth of this is about the secondary growth in the dicot stem. During the secondary growth, some of the cells rupture and dead. So if you observe, rupture and dead. The bar is a tissue which is present in the dicot stem. Here the all tissue exterior to the vascular cambium including secondary phloem, is termed as bark. It refers to a number of tissue types like periderm, secondary phloem. Periderm and secondary phloem together, it is called a bark, and bark formed early in the season, it is called early or soft bark, towards the end of the season called late or hard bark. Again, the plant contains the lenticels. Lenticels is nothing but in a woody plant, there is a lens shaped opening. If you observe, there is a lens shaped opening at certain region of cork, which permits the exchange of gases between the outer atmosphere and the internal tissue of the stem. This lenticels again has many cells within the lenticels, that is epidermis, complementary cells, cork cambium, secondary cortex. This is about the lenticels. Next, hood is divided into two types: spring hood or early wood, autumn wood or late wood. Spring wood, the wood formed during spring season is called spring wood. Autumn wood, the wood formed during autumn season is nothing but autumn wood. The difference is cambium is very active during spring wood and produces a large number of xylary elements having vessels with wider cavities. In the autumn wood, cambium is less active and forms fewer xylary elements and have narrow vessels. And in the spring wood, it is lighter in color. Spring wood, if you observe, it is lighter in color and has lower density. The mass will be less in the spring wood. Autumn, darker in color and higher density. Each ring or a concentric ring represent a annual ring. This each ring represents the age of the plant. So this age of the plants is the study of the age of the plant is nothing, called as dendrochronology. Next is the heartwood and sapwood. The center of secondary xylem. Heartwood has in the heartwood, at the center it has a secondary xylem. Sapwood, at the peripheral it has a secondary xylem. It comprises dead elements with highly lignified walls. Sapwood is lighter in color because no lignin deposition. Heartwood, it does not conduct water but it gives mechanical support to the plant. It is involved in the conduction of water and minerals from root to leaf. So heartwood, as I told, dead elements with highly lignified walls, and even this heartwood has a substance like resins, tannins, gums, aroma substance and essential elements, and this region which contains the substances, it is durable and it is resistant to attacks of microorganisms and the insects. This is about heartwood and the sapwood. Next, secondary growth in dicot root. The same mechanism occurs in the dicot root. Only difference is here the pericycle. Here the pericycles form a wavy cambial ring, phloem, xylem, pericycle. All this combine to form a ring, and later it divides and form a cambial ring, and again the same process occurs in the dicot root also. What are the process occurs in the dicot stem? Same process occurs in the dicot root. Here remember, pericycle cells form wavy cambial ring, whereas in that medullary rays which is present between the adjacent vascular tissue. This is what about secondary growth in the dicot stem. Now let us solve some of the questions. Meristematic cells are characterized by option A, secondary wall and centriole, option B, thin cell wall and no intercellular space, option C, thick cell wall and small nucleus, large vacuole and well developed plastid. Before that, let us summarize the characteristics of meristematic tissue. So it has isodiametric with the thin cellulosic cell walls with no secondary cell wall. So it does not have a meristematic does not have a secondary cell wall. They divide rapidly and there is no. So this option will be it does not have secondary wall. So this option will be cut off, and they divide rapidly and there is no stored food in that cell, there is no stored food. These are compactly arranged with no intercellular space. So no intercellular space, maybe this may correct. Let us see another. They have a dense cytoplasm with a prominent nucleus. So this is also wrong. Prominent nucleus, it has a prominent nucleus. So option does not have any plastid thing. Option B is the correct answer. It does not have any intercellular space. So the option B, it has a thin cell wall and no intercellular space. And the next is the apical meristome is present only in radicals, only in taproots, only in adventitious roots, in all roots. Apical meristome is nothing but at the tip of the plant a meristematic activity present, that is called the apical meristome. So all these three has the apical meristome activity. Option is in all the roots. Next is chlorenchyma cells are. Before that, let me explain. Parenchyma has two types of cell, chlorenchyma and aerenchyma. Chlorenchyma cells have chloroplast within the cell. Aerenchyma cells, air cavity present in the cell. So if you look at the option, chlorophyll containing phloem, it is wrong. Chlorenchyma containing sclerenchyma cells, it is also wrong. As I already told, it is a type of parenchymatous cells. Chlorophyll containing epidermis, wrong. Chlorophyll containing parenchyma cells, it is a right answer. Next is the collenchyma occurs in the stem and petioles of collenchyma. As I already told, it helps in the photosynthesis. It helps in the photosynthesis, storage and all. But here they are given xerophytes. Xerophytes, it is present in the where there is no sufficient of water. So collenchyma, so as xerophyte is a wrong answer. Monocotyledonous plants, option C, dicotyledonous herbs, hydrophytes. Option C is the dicotyledonous herbs. Our answer is the dicotyledonous herbs. While explaining the stem, I have differentiated dicot stem and monocot. Hypodermis is made up of collenchyma. So remember the anatomy of the all three, that is stem, root, leaf, so that you can answer correctly. So collenchyma occurs in the stem and petioles of. So remember, while explaining dicot stem, I told hypodermis of dicot stem is made up of collenchyma. So option C is the correct answer. Next, dash provide mechanical support to the growing parts of the plant. So it is a direct question from the textbook. Without saying any other option, you can say it is a sclerenchyma. Option 6. Which of the following is not true about sclerites? These are groups of living cell. So sclerites, it's a group of living cell found in nutshells, guava pulp and pear, also called stone cell, form sclerenchyma with fibers. So option sclerites is a type of sclerenchyma. So sclerenchyma is not a living cell. So option A is the correct answer. All these three is right answer. Pits are found on the cell wall due to lack of primary wall material, B, middle lamella, C, secondary wall material, D, all of the above. So before that, let me explain what is this pits. Pits are the thin region of the cell wall through which the adjacent cells communicate or exchange fluid to the other cell. So observe, this is a cell, adjacent cell. So within this it has a pits. This region is called the pit. Through this region, adjacent cell gets the water or any other material. They exchange the fluid to the other cell. So this pit is made up of three parts: pit chamber, pit aperture, pit membrane. Pit aperture form opening of the pit chamber. Pit membrane composed of primary cell wall and middle lamella. So pits are found on this primary cell wall, middle lamella. Option A, option B, both are correct. And pit chamber is hollow part on which does not have secondary layer of cell wall. So it does not have secondary layer of wall. Answer is secondary wall material. The basic difference between a vessel and a trachea is. What is the basic difference between vessel and trachea? So the basic difference are this tracheids and vessels are tracheary elements. These cells are devoid of protoplast. Both doesn't have protoplast when mature as they become non-living cells with elongated lignified. This is the similarity. Both have same type pits arrangement. Main difference of vessel is perforation at the end plates which make them a tube-like long structure, while tracheids do not have end plates. Vessels as a end plates, whereas tracheids doesn't have a end plates. So the option C is the correct answer. Next, sieve tubes are best suited for translocation because they, option A, possess no end wall, option B, possess a broad lumen and perforated wall end, are broader than long, and possess border pits. Option here, sieve tube, before that, sieve tubes members are only found in angiosperm. They have pores which are larger and called sieve plates. They occur end to end forming sieve tube, which also better translocation of food. It has a answer is it has a broad lumen and perforated wall end, hence it is best suited for translocation. Next, an organized and differentiated cellular structure having cytoplasm but anucleated. So, while explaining the phloem, as I already told, vessels, it has a xylem, parenchyma, it is also has nucleus, sieve tubes doesn't has a nucleus, enucleated, trachea also has nucleated. Companion cells are not found in where it is absent. It is present in angiosperm but it is absent in pteridophyte and gymnosperm. Both A and B is a correct answer. A piece of wood having no vessels must be belong to. If you remember the xylem, I was explaining about vessels. Vessel absent in the gymnosperm. If you look at this, teak is a angiosperm, mango is also angiosperm, palm is also angiosperm, pine is a gymnosperm. So answer is pine. Next question, epiblema of roots is equivalent to. So in the tissue system, I told epidermis is termed in the dicot root, but epiblema is called epiblema is termed in the monocot roots. So epiblema of root is equivalent to epidermis. Epiblema is the outermost layer in the root system. Epidermis is outermost layer in the dicot root. Ground tissue system consists of. So remember, ground tissue system consists of ground tissue middle, that is except epidermis and vascular tissue, all other tissues constitute as ground tissue, that is option D is correct. Hypodermis, cortex, endodermis, pith, because it does not have vascular tissue, it does not have epidermis. So answer is hypodermis, cortex, endodermis and the pith. The next is a bicollateral vascular bundle is characterized by bicollateral phloem being sandwiched between xylem, transverse splitting of vascular bundle, long-term splitting of vascular bundle, xylem being sandwiched between phloem. So bicollateral, what is bicollateral? Bicollateral, it is a type of conjoint vascular bundle. In the conjoint vascular bundle, phloem, xylem present between the between that it has a cambium. But here at the either side it has a phloem and in between it has a xylem. Option A is the correct answer. Phloem being sandwiched between xylem. Which one contains vascular bundles in a ring? Which one contains vascular bundles in ring? Which one? Option C. Option. If you observe the anatomy of the monocot and dicot root, all the A, B and D are monocot root. But option C is dicot root. In the dicot stem only you can observe ring form of vascular bundle arrangement, whereas in monocot root it is scattered form. So option C is the correct answer. Next is identify the type of vascular bundles. So first one is radial, second one is it has a cambium, so conjoint, does not have cambium, conjoint closed. A third one is it has a cambium and xylem phloem, conjoint open. Option A is the correct answer. Next is pericycle of roots produces, it does not give mechanical support. It produces lateral bundles, vascular bundles, adventitious spurs. So if you remember the morphology of flowering plants, I had explained lateral roots is the roots which is produced from the pericycle. Passage cells are thin walled cells found in. So while explaining dicot root and monocot root, in the endodermis, some of the cells has a waxy layer, that is called Casperin strips. Some of the cells lack waxy layer, it is called passage cells. So where it is found, it does not found in the phloem elements, test of seeds, it does not found in the test of seeds. Central region, it does not found in central region. It present in the endodermis of root, facilitating rapid transport of water from cortex to pericycle. Next is a major characteristics of the monocot root is the presence of, what are the major characteristics? Without seeing other option, you can say monocot root. So don't be confused. Monocot root, don't put it, write as scattered vascular bundle. Scattered vascular bundle present in the stem. Vascular without cambium. In the monocot root, it has a radial vascular bundle. Anatomically, roots differ from stem in having, how it is differ? Anatomically, roots differ from the, differ in stem in having what it has, parenchyma present, both stem and root. So parenchyma, answer is correct. Pith, both present in stem and root. Exarch xylem, root is exarch, stem is endarch. What is exarch? In the exarch, metaxylem towards the center, protoxylem towards the periphery, whereas in the stem it is endarch. Metaxylem towards the center, protoxylem towards the periphery. This is the main difference, not a pericycle also. Main difference is exarch in the root, endarch in shoot, stem. So next is the bulliform cells take part in, it is also a direct question. So answer is the curling of leaves. Cambium activity is highest in which season? So in your book you have only 2 seasons. So option B, D is, just forgot about option B and D. A, C. So during the spring season only cambium activities is high. Secondary xylem and phloem are led on by, vascular cambium. Due to the vascular cambium only secondary xylem are formed. Whereas next is the periderm includes secondary cortex, nothing but secondary cortex is also called phylloderm. Cork is nothing but phylum. Cambium is nothing but phyllogen. All of this. So remember, secondary cortex is also called phylloderm. Cork is also called, that is cork cambium, phyllogen. Cambium. Cork-cambium. Cork is phylum. This is phyllogen. So all this three includes Peridot. The next is heartwood differ from sapwood in heartwood. How it is differ? Heartwood is differ from heartwood. Presence of rays and fibers, absence of vessels, and parenchyma having dead and non-conducting elements being susceptible to pest. Differ from having dead and non-conducting elements. Option C is the correct answer. Heartwood differ from sapwood being it is not susceptible. It has a resistance power to pest and pathogens. So option D is wrong. Cells towards pith mature into what? Cells towards pith mature into secondary xylem. Match the following. So before answering, you have to observe right column and left column. In the left column, you have a different tissues. In the right column, you have a different option. That is, look at this option. If you look at this option, you will notify that is function of this tissues. So what is the function of stomata? Helps in the transpiration. So for the stomata, option 4 is the correct answer. Bast fiber, it is a dead phloem fiber. Vessels, what is the function of the vessel? Xylem vessel. Xylem vessel found only in angiosperm. Correct answer. Trichomes, it is a epidermal hairs on the stem. Lenticels, it helps in the gaseous exchange in the woody tissues. So if you see option A 1 4, option C also has 1 4, whereas second 2 in the option A it has a 2 2, option 2 it is the right answer. Option C is the right answer. The next comes the anatomically jute fibers are, if you observe the option vertical fibers with pith fibers, you never noticed this words in the textbook, that is vertical fibers, pith fibers. So for you to confuse you, that given this option. Xylem fibers, it is a type of cell which is present in the xylem fibers. So jute fibers present only in the phloem fibers. The distribution of stomata in dorsiventral is, so without saying, less stomata in the upper surface, more on the lower surface. This is the right answer. So, thank you.